Good afternoon, friends. Welcome to this session. And we are fortunate to have a great faculty for this session. Dr. Arun Bhatt. Bhatt sir is very well-known mentor and researcher in this area. I think most of the clinical researchers, they are trained by Bhatt sir. He is working as honorary editor of the journal, very well-known journal in this area, that is Perspective of Clinical Research. And he is well-known as a consultant, guide for clinical research and drug development programs since last uh, more than 30 years. Based in Mumbai, Dr. Bhatt is involved in various training programs of several industries and government platforms. Welcome you to this session, sir.
Good afternoon. Thank you, Dr. Girish Tillu and I think friends from the university. It's my pleasure to share with you some of the experiences which we've had during COVID, and then of course what has happened to clinical trials. And as Girish and I were talking, please feel free to ask questions whenever you want to in the chat box, and we'll start answering questions as and when the questions come up. So you don't have to wait till the end. And some topics are little heavy on statistics, so I've avoided statistics, but I've tried to give you concepts. But overall, I'm trying to give you an idea of what happened in COVID and what probably is now a focus of the new research. In fact, just about 2 weeks ago, there was a panel discussion on post-COVID scenario in terms of drug development, and I think everybody talked about what is new normal. And most of the people from industry said, "Well, it's more of new abnormal," because with what we did in COVID, no, people have thought that this can become the new normal in terms of doing clinical research or clinical trials. Well, that's probably overstating things. So I've tried to put down the title is post-pandemic paradigm shift. So there is some shift happening in the way we do things, but it's not yet widely accepted. One of the problems has been that what is it that works? No, I mean we have all been kind of wondering that everything that we tried out in clinical research has worked, but actually the news was so much overwhelming in terms of what they're telling about clinical results. And even today, I think the headlines come from media. In fact, more like if you shut your eyes to media news, you might help. So what was the challenge in pandemic situation? First of all, we want a rapid deployment of treatments. I mean, there was no treatment available. If you look at 2020, March, almost now 2 years back, when the whole problem started, there was no treatment available. Of course, there are a lot of theories about what could work and what could not work, but there was nothing readily available in terms of tried and trusted kind of treatment. Of course, even the trusted part is even today questionable. But the second challenge was, is it adequate scientific scrutiny? Is it really scientific? And this is where whole struggle has been for the pandemic situation. Just to give you some idea of what happened in pandemic, almost 5,000 plus trials were done, and of course they are still probably ongoing for COVID-19. It's a very old slide now, and you see most of them were interventional trials. So this is you can see, okay. And if you see most were phase 1, 2, 3, 4 trials. Okay. But in most of these trials, there was a problem of doing a good scientific trial, and just to give an idea of how things were, BMJ has been carrying out meta-analysis of ongoing trials, and in that they are actually trying to look at what works, what doesn't work. But more than that, they have also looked at the quality of trials. No, so this is the latest information from March 2021 paper. Uh, they have looked at about 196 trials on COVID treatments of more than 76,000 patients, and they found that 78% of trials were of high risk of bias. Only 22% were of high quality. So that's a problem of pandemic research, that whatever data we have got right now is probably comes with a lot of bias in terms of how the design is planned. And basically the problem has been randomization or deviation of the intentions. So the biases common have been randomization, departures from intended intervention. For example, I mean, just to give you a simple example, in COVID, I think as a doctor, if you are doing a clinical trial, it's an open trial because it's difficult to blind a new therapy. So you are doing a style of standard care versus another therapy. So you have an add-on therapy. So standard care has been now changing over the last 2 years. Now it's, of course, now today we can say it's oxygen, remdesivir, and these are the two, I think, kind of mainstays of the treatment. So you will have a trial of remdesivir, oxygen, vis-a-vis standard care plus remdesivir plus oxygen. Now here, the doctor who's in the trial can have a bias. If he or she feels that okay, the two are enough, steroids and remdesivir, they may not randomize the patient to a new drug. But the other way around, during early phase of clinical trials, people are so desperate that whatever they believed in, they would try out. So they might mess with randomization. Okay. Outcomes data I've been missing sometimes. Measurement of outcome. A lot of outcomes are measured now, which are more based on symptoms. I mean, maybe we can talk more about it, but when we saw a lot of studies being done in mild COVID, that was the question. In mild COVID, everybody is going to recover, even without any specific treatment. Most would require no symptomatic treatment and just managing fever. So it will recover. But if you do a trial in that and so no, it's going to recover totally because of new drug. Well, that's a wrong message. And ultimately selective reporting of results. So this happened in very often in even Indian trials. I remember in favipiravir study, which was talked about in the first year. Some of the data which are given, I think probably were highly selective data. Talking about rapidity of reduction of the rapidity of conversion of positive to negative RT-PCR. Now that kind of headline was hiding the fact that even without a treatment, the test can become negative. So what has happened overall in terms of different designs? First of all, we'll talk about real world studies, then master protocols, which are also adaptive designs in a way, and digital trials. Okay. So first, let's talk about real world studies. I mean, real world studies are basically nothing but if your clinic or hospital has its own data set, which is not collected for a clinical trial purpose, it can be used to derive some information. This is the official definition. Data related to patient health status and or delivery of healthcare routinely collected from a variety of sources. I'll just switch off my video to save some bandwidth. Okay. Yeah. And this could be from multiple sources. I mean, as I already about we can have, let's say, clinic, hospital. These are known sources. But you can also know data coming from insurance companies. You can also have data coming from laboratories. So there are different places from where the data can come, which is outside clinical trial. Though during pandemic, it is said that these studies have potential to provide valuable, rapid, actionable information. Most of the work which you have seen in pandemic about the epidemiology or the vaccines is all real world data mostly. To better understand natural history and course of disease, and to answer urgent questions on effectiveness and safety of treatments and health economics and outcomes research. Obviously, most of the work was done in COVID was in this area, and I'll just show you some examples of this. Now, what is the challenge of this? If you look at in real world evidence in pandemic situation, we have so much data on diagnosis, risk factors, for example, age, immune status, drugs which are immune modulators, okay, diabetes, hypertension, clinical information, symptomatic information, right? From simple, COVID cough to anosmia to now post-COVID syndrome, management data, prognosis, that means long-term follow-up, exposure and outcome. The exposures have been mostly here. Either you have the disease and the outcome, or it would be a treatment and the outcome. But you are in the real world. When you are in real world, I think what about high exposure population? For example, doctors were high exposure. What about people with elderly who are high risk with comorbidities? So these are additional issues along with the data. But the evidence requires data, first of all, quality of data, planning very well, good studies with design to make it valid, and then it becomes utility and real time. Okay, in real time means as it happens. I mean, and unfortunately, I think just to give you an idea of how things happen so fast. This is a UK study done in the healthcare database of general practice, which the UK healthcare collects a lot of data of that nature. This was a study done published way back in May 20. As you can see, within 2 months of the COVID becoming a really florid epidemic, population about 17 million adults, and they use primary care electronic health records. So this is one reason which people talk about no electronic health records. If you want to do real world data quickly, you require electronic health data, because our current method no working with no paper based case record forms may be laborious and maybe not really effective. Within 2 months of the treatment of the epidemic, 1st February to April, they did the study. The primary outcome was death in hospital among people with confirmed COVID-19. This was a cohort study. It's like a registry study. They adjusted for age and sex and other covariates. It's very important that in a real world study, you have to adjust the covariates, otherwise they can no influence the results. And this study also had ethics committee approval. Now, what did they find? This was the first study which showed that no people, the death was higher, male sex, greater age, deprivation, poverty, diabetes, severe asthma, obesity, other conditions, and more in Black and South Asian people. So this was one of the first studies which showed that this can happen. The later studies which came off with vaccines, this is the study comparing no two populations, a different way of doing a real world study. This is the Israeli study where this is insurance database. I mean, and we don't have insurance coverage in India, which is widespread, and that's why we don't have so much data. But in Israel, virtually everybody is covered by insurance, private, maybe or public. And one of the largest insurance companies had about in this study about 12 lakh, 1.2 million patients, of which half were on vaccine, other half were not on vaccine. And they followed them up to show that those who were on vaccine, 2 doses, we could show reduction of risk of infection, illness, hospitalization, severe disease and death almost by 80 to 90%. This was kind of supporting data on the clinical trials of vaccine, which claim to have almost 95% reduction of infections. But yes, the real world evidence and data depend on quality of data. The data quality should be accuracy, relevance. It could be measurable. It should be you should be able to source with data verification, and very fast, rapid. If you have multiple site studies, should be consistent. The design requires good attention, because these are not standard just case series or collecting some 100 cases, but you have to put a design which is largely coming from epidemiology, like cohort, case control, or I think or the surveys which you do. And the conduct has to be again ethics, documentation, analysis, and interpretation. Now, there are standards for this, and without these standards, we cannot talk about real world evidence. So I that time wrote an editorial in the journal which I'm editing right now, because I saw a lot of studies which had come from world over, from USA, from China, from UK, Israel, but very few studies or no studies from India of this nature. We did have some small publications. I remember a series of publications in Journal of Association of Physicians of India, about 100, 200 patients here and there, but they could not give much information. They're more like descriptive studies, with some data on the disease and the risk factors. So this kind of studies required patience of academic researchers during trying times, ability of detailed information on patients. Because if you want to conclude the data on, let's say, drug effectiveness, you require outcomes data. Ability of high quality electronic health records. This is the way it moves fast. Rapid access to large medical data sets to multi-institution, multi-country collaboration. Now, these are large numbers. If you're going to do a study of real world evidence in about 100 patients, it will not give you much data, but you might require 1,000, maybe require 5,000. Now, you want to collect them very fast. Multiple hospitals or clinics have to come together. This is what most studies have shown. I mean, I have seen in most studies, there are 20, 30, 40, sometimes 40, 50 sites coming together. Technical support for advanced data analysis for extraction of data. So once you have data, you have to extract and do analysis, which is quietly done by statistics. Open access to data for research. This can only happen if multiple hospitals have or even the government has open data for research. For example, the Israeli study I cited for vaccines. The Israeli government every day puts out data on the infection rate and the hospitalization rates. It's available to everybody, and you can easily do a research on it. In contrast, in India, if you want to do research on even government data, even today you can't get data. People have been commenting that in India the control is with the government of all the data, and you can't get data to do good research. Also management of ethical data privacy and security. Now this data actually are belonging to patients. They are generated in the hospital. So the first right is of the patient and of course of the doctor. So you can't just use the data commercially, or the company can't use it without no having adequate ethics and data privacy issues tackled. And finally, these are requiring advanced statistical methods to handle issues. So that's the first session of the talk. So if you have any questions, we can address them now. Else I can go to the next topic, adaptive designs.
I think, sir, we can have questions at the end of the session.
Okay.
Okay.
Let's go to adaptive designs. I think these are not new actually. The designs, I mean, I remember even writing an editorial way back in 2007, 8 in one of the ICMR journal, IGMR. So this is not a new concept. But what has happened now, I think the concept has come into limelight because of the need for doing fast and well research in the pandemic times. So it is definitely not a simple clinical trial as we think about. I know kind of this just talks about how it is, just how do you do milkshake now by kind of shaking the cow. But today I think you don't do that way. Today you require a very high level of sophistication of doing it to a mixing. So same way, the standard clinical trials and adaptive have this kind of difference. So what's adaptive design? Now you can see even as late as 2019, Food and Drug Administration, that is FDA of USA, has given a full guideline and the design. So although these are not new, but they still remain very current because of the changes in the way things can be done and way it can be creative. Clinical trial design that allows for prospectively planned modifications. See the word modification to one or more aspects of the design. You can modify some, which are what we'll do a little later, based on accumulating data from subjects in the trial. See, this is what is important. In traditional trial, we analyze the data after the trial is over and all the data are put in. But here, we start doing the analysis as the data are accumulating, because that is going to influence the decision of modifying certain aspects of trials. Just to give some idea pictorially of clinical trial design, I would just request you to see this website. Although this is a website for patients, European Patients Academy, UPAT. It's a very nice website, simple website for patients to understand, clinical research and drug development and therapeutic innovation. And they put very simple designs. Now, for example, this is one of the examples for adaptive design now. So you have, let's say, a population. You randomize the two people group, and then you decide the endpoints in advance. No, this is actually for cancer. So progression-free survival and overall survival. And then you are randomizing. See, this is a single drug, and this is actually there are two drugs here, compared. And at each point, you start looking at the data. So if there is a potential that the drug is effective, that will move forward. If it is not, it will stop here. So this is the way. So it can stop early any group depending on the data and its outcome. If it's negative outcome, you stop the I think work here for this group or for this group. We don't want to subject more patients to ineffective treatments. Another one which I think we'll see in more detail, which is now coming to what they call now multi-arm, multi-stage design, which is now also part of a lot of master protocols. Here you can actually compare multiple therapies. Anyone. So you start with one control arm, and there are 3 or 4 different new drugs, no arms, and then you can have analysis which is interim 1, 2, right? So at this analysis plan, again, you judge which of the therapies has shown efficacy. The one which shows efficacy goes further. The one which doesn't show stops here. So that way you are able to remove this arm. And same way you are able to add more arms depending on when there's a potential purpose. So if you see at the end of this phase 3, and this actually also has been used very often in this what they call phase 2-3 design. That you start in phase 2, multiple therapies, and then you see only these 2 therapies are going forward into phase 3. They've stopped the other therapies which probably are ineffective. This is actually another one is seamless phase 2 design. That means you are here randomizing a group of people in phase 2 to different 3 groups. Again, you're looking at cancer as an outcome, progression-free survival, and see what has happened now. See, this therapy has stopped, okay, and these two have moved forward to efficacy. Okay, these are some examples. So now we'll see more of it. Specifically, what is adapted. So first of all, there are two ways of adapting the design. First of all, you don't compare the data in different groups. So you do a blinded analysis, because one of the challenges of all these studies is that if you don't blind properly, and if it's unmasked, a lot of bias will creep in, and then you will probably end up selecting ineffective therapy. So you can do a blinded analysis or pooled analysis. That means you put the whole group together, analyze, and then you try to analyze blinded sample estimation. That means you whatever comes out based on the available data, you might decide to kind of change the sample size. But very often we do it what of comparisons of multiple drugs. So one is actually group sequential design. Then second is adaptation to sample size, patient population, treatment arm selection, patient allocation, endpoint selection, and multiple features. So all these can be adapted as the study continues. Okay. Obviously, this will require a very high level of planning and statistics. Just an example of what are the different studies one can do. This is from a recent article in BMC Medicine. And the design is this. For example, group sequential. Include options to stop the trial early for safety, futility, efficacy. That means you start in the group, and as I showed you earlier, you can stop the trial early if it's unsafe, or you have not reached the number, or if it's not effective. Sample size re-estimation. Adjust sample size to ensure the desired power. I mean, those of us who are used to this word power, when you plan a study, you plan a study based on statistical considerations. No, so you look at the power of the study to really give valid data. But you find when you start the study that the power is not what it was. And then you might change the sample size. Okay, multi-arm, multi-stage. I showed you explore multiple treatments, doses, durations or combinations with options to drop losers or select early. So that means ineffective therapy is stop, or you start selecting effective therapies early. Population management. Once you know that a particular population is going to benefit more, then you might start changing the criteria. So narrow down recruitment to patients more likely to benefit from the treatment. So you don't kind of the other patients who are not benefiting maybe kind of withdrawn. Adaptive randomization. Shift allocation ratio towards more promising or informed treatment. Now, this is actually a very interesting thing, that usually we assume that okay, if you have a five arms in a study and 100 patients, and you randomize, you will be having 20 in each arm. But here it's that if something is more promising, then that particular arm might have more. So that means although it's 5 arm, you might have 3 arms with 10 each, and then 2 arms with 20 each. Just a minute. Yeah. Just to continue here on this adaptive randomization of promising results, adaptive dose ranging. Again, this is more often relevant to phase 2. And I mean, you might be aware that in phase 2 of clinical development, after we have finished the first phase in human healthy volunteers, we come to phase 2. We are trying to find out which dose is going to work in patients. So obviously you try multiple doses, and here I think adaptive design will try to go towards more promising or informative doses very quickly. So we talked about seamless 1-2 combine safety and activity assessment into one trial. This is what actually was done in many of the vaccine trials last year. They started phase 1 in healthy volunteers in a small number, and the later part of study was phase 2 once they reached the dose which was showing antibody levels in the in the uh, after the vaccine. And similarly you start doing the phase 2 trial, so the larger trials in vaccines after the initial acceptance of dose were more of phase 2-3 trials to look at what happens in long term. So what are the benefits of this adaptive design? First of all, it's statistically very efficient. Greater chance to detect a true drug effect than a comparable non-adaptive design. This is what is expected, that since you are using multiple different therapeutic options, you have control, and you are using very advanced statistics, you can get a true drug effect compared to a non other designs. ethical consideration. No, this is very important because no, we today know when you are doing a clinical trial, we don't know. I mean, we actually try to complete the trial in everybody. Whether they whether the even if the therapy has failed, the trial continues. No, you don't know the results till the end of it. While here in adaptive, you can stop the trial early and reduce number of patients exposed to unnecessary risk of an ineffective therapy. And similarly, you can quickly take a effective therapy and start putting more patients on it. So from the patient's viewpoint, adaptive designs is more ethical because the chance of getting a less effective treatment and chance of getting a more effective treatment both are much higher. Okay. Improved understanding of drug effect. You can answer multiple questions in the same design. You can put multiple questions. And because of this flexibility to modify and increase the probability, I think both the patients and also the investigators are very happy because they have option to modify the trials. But today, I think in a large phase 3 trial, which is done globally in companies world over, there's no scope to modify the design halfway through. No, adaptive has a chance to change it. Obviously, the change will not come because a doctor or patient wants it, but because the data suggests that well, there is a need to change the sample size or the dose or the treatment. But yes, there are limitations. These studies, they look very attractive, very creative, very innovative, but these are not everybody's cup of tea. In fact, I've yet to see adaptive design being done regularly in India. I mean, I've not seen much. No, I mean there was some discussion of that in vaccine trials, but when I started searching the literature, I didn't find much of it. But yes, internationally, academically, and as I'll show you one example of what has been done in I think I told about vaccine trials. Similarly, I think there are therapeutic trials in COVID. Some of the examples I'll show you how they've been done. So requires specific analytical methods to avoid increasing the chance of erroneous conclusions and introducing bias in estimates. That's the first thing. I mean, since you are able to do multiple analysis interim, you can have bias because you would be tempted to stop or start things. Pre-planning adaptive design modifications require more effort at design stage, leading to longer lead times between planning and starting a trial. Now, for these trials are not done by one or two hospitals. No, I mean they are done by for example, if you see the master protocol, it was done about some 100, 500 British hospitals all over the country. So obviously the planning, adaptation of that design, all this is going to take longer than a standard trial which you can make a protocol and go straight to ethics committee and get approval and start in maybe 2-3 months time. But here it can be much more because everybody has to agree to one approach. Additional logistical challenges to ensure appropriate trial conduct and integrity. So when you have multiple centers, multiple options of changing the design in between the samples, unless you ensure oversight and conduct, it might happen that your trial quality may not be great. And finally, interpretation of results. I mean, I'm just telling you very simplistically something not effective or effective, but I think these are all very subtle things, in clinical trial. For example, in cancer setting, progression-free survival, usually is in terms of numbers, in months. So what is the good number? Is it 9 months survival? Is it 12 months survival? 6 months survival? One doesn't know, no? Continuing further, logistical challenges to ensuring appropriate trial conduct and integrity. So what happens? I mean, so you require data to come in very regularly. So it's not going to happen now with the standard paper-based trial. I mean, when I wrote the editorial, I remember that we had to look at populations, the data. It does be electronic data. The data should be coming in as soon as the visits are completed. The data should be cleaned up by the data management and analyzed as required at the appropriate time, ongoing way. And the data are fed back into the system. And I think you require very often committees to sit and look at the data to make decisions about whether to trial should continue as it is or trial should have change of the design, because whatever is expected has to be approved by somebody else. So logistical challenges to ensuring appropriate trial conduct and integrity, limit access to comparative interim results may be complex and cost to the trial. So this is what is the crux of the matter. Interim results of the efficacy or safety which are generated in this trial should be known to only a few people who are going to take decisions about how the trial should move forward. But if large number of people come to know, the doctors come to know, they might be tempted to ask for treatments which they feel are working, while it should be happening at the level of central level. Ensure high quality interim data are available in timely manner. See, timely manner. So these studies require a lot of planning, and a lot of, for example, if you look at the data, which I'll show you on the master protocol, how it happened, it was so fast, no. Okay. So you require, see, up to date, reliable results now. This is the this is the heart of the study. So this can be done provided all the sites are ready, up and running, and I think there is a central agency which is able to ensure this conductor integrity. Any questions here?
So there is a question, can variability in atmosphere impart multicentric adaptive trials while changing sample size for more effective therapy? So there can be many variables affecting outcomes.
Yeah, that is taken care of in the basic randomization and blinding. So those principles are not changing. Adaptive doesn't mean that you don't randomize or don't blind. That part is there. So when you randomize and blind, you are going to control and compare variables which are well known to affect the final outcomes. So I mean, the standard demography will be comparable. The disease status will be by and large comparable in the groups. So that's probably unlikely to affect the kind of data directly. But yes, this is always a question that whether the variability in the baseline data or demography can affect. It can always but I think that is less likely because you are planning a standard randomized double blind placebo control kind of study, trying to balance these factors.
Thank you.
Any other question? Okay, let's go to the next one now. This is actually master protocol is a one form of adaptive trial. So it's not really different, but the principles are now applied for a specific purpose, and I'll try to show you what I have learned. But I mean, I've not seen a trial being done in India so far. Although I think there were a lot of talk about doing a ICMR such a trial for COVID. What they call I think solidarity of WHO. But I have not seen this being the way it was done systematically for recovery trial. So frankly, although we all know trials, when it comes to master protocols, we have to learn a new way of doing a trial. So like in this cartoon, instead of taking the tablet for slimming, it's more like putting it on the table and then picking up one by one. So what's a master protocol? One overarching protocol designed to answer multiple questions. Now, what these questions could be, we have to see it later. So may involve one or more interventions in multiple diseases or single disease. So you can have in same study multiple diseases being tried out, or one disease as defined by current disease classification with multiple interventions, each targeting a particular biomarker defined population or disease type. Now, by and large, this concept of biomarker defined population is largely there in cancer. I mean, those of you who have seen maybe cancer patients in kind of a family, or maybe in clinical trial. I think now there are a lot of biomarkers are used to define subpopulations. Now, for example, I'll just give a simple example of breast cancer. I think some of you who are senior might know that breast cancer. When I started my career in medicine, for breast cancer, we talked about only radical mastectomy, remove the breast, that's all. And then it came remove the other breast also, or the lymph node also, because we couldn't do much more than that. Then came chemotherapy. So chemotherapy became the standard, and obviously then it was whether chemotherapy can reduce the size of the cancer. So it was like adjuvant chemotherapy. That means you give chemotherapy and then do surgery. That was the thing. But later on, they started finding out that there are subtypes of breast cancers which might improve with the particular therapy. So when I was in Novartis, the hormonal basis of treatment of cancer became very popular. So we did a trial of a drug called letrozole, which is aromatase inhibitor. So which I think was working in women with breast cancer, advanced breast cancer, usually phase 3, stage 3, who were positive for either estrogen or progesterone receptors. So ER/PR positive. So in that subgroup, we started giving the hormonal therapy, and I think there were good results coming up of improving the efficacy. This was not everybody. I mean, some women whose tumors are showing positive. Then came another biomarker called the HER2/neu. Now HER2/neu is I think a genomic proteomic biomarker, which is again, if it's positive, then this there are newer therapies which are more monoclonal antibodies are working there. So you see the biomarkers now. So if you see today, when you look at breast cancer, if you ask a patient of breast cancer who goes to oncologist, they'll typically do all this. They'll do a standard biopsy, then they'll do a subject biopsy to receptor estrogen progesterone. They look at HER2/neu, and now I think there are more things coming up as that things change. So biomarkers are now defining subpopulations which potential have a better outcome possible. Now there are three types possible: umbrella, basket, and platform. Now obviously these are not technical names; they are more like pictorial presentations of how the trials look. So 2017 article of NEGM talks about umbrella trial to study multiple targeted therapies in the context of a single disease. So it's more like I mean both maybe the COVID trials some of them are like this or for example in cancer they do multiple trials single disease cancer and then multiple therapies basket to study a single therapy in context of multiple diseases. So here you kind of one, and this happens very often in immune drugs in cancer, that many new drugs in cancer might work in multiple indications. Now the challenge is you can go on doing multiple different trials. For example, let's say an immune drug works in lung cancer, works in breast cancer, works in head and neck cancer. Now you would end up doing three trials. But here they're saying that no, you can do actually one therapy in three trials in the same kind of design. And finally, platform trial to study multiple targeted therapies in the context of a single disease in a perpetual manner. See, this platform, it continues to have the trial. The trial doesn't stop. As you learn more and more, you can add more therapies. With therapies allowed to enter or leave the platform on the basis of decision algorithm. So therapies can be stopped or continued or added as it happens. And this was, I think, the trial which was done, this recovery trial in UK by Oxford. And I think when we talk today about that dexamethasone is the drug which works, I think this was a major contribution of this platform study from Oxford. So umbrella trial evaluates various, often biomarker-defined subgroups within a conventionally defined disease. As I told you about, that you can have a breast cancer patient with multiple subtypes. Patients with the disease screened for presence of a biomarker or other cases and then assigned to a stratum on the basis of results. Multiple drugs are studied in these various data. Design may be randomized or use external controls. So, I mean, here in this case, you can have a control within the design, but if you don't have that possibility, you can use a available control in the published literature as a control arm. So this is the umbrella trial. You see, this is a picture of umbrella now. It's a single disease. You screen for targets. So you have biomarker 1, biomarker 2, biomarker 3, and then there are different therapies. If you see. Okay. This was an example of a I-SPY2 trial, which is done in breast cancer. They told neoadjuvant treatment of locally advanced breast cancer. Now, neoadjuvant is like in the surgical setting, you add treatment, okay, and specific. At consents, a new patient's participants breast cancer classified into one of the several 10 molecular subtypes. See, there are 10 subtypes here. I told you about 2 or 3, but there are more. Response adaptive randomization. That means based on the response, whether the therapy is working or not, you start assigning patients to most promising treatment or combination of treatments in the respective 8 genetic breast cancer subgroups. Sufficient number of patients assigned to the standard care. So you require a control arm with standard adequate number, and shared use of control patients across treatment. So this will become the control arm for all of those 8 different subgroups. Now endpoint was assessed at the time of surgery. Pathological complete response with tumor at surgery. This is supposed to be endpoint. So whenever you reach the endpoint, you decide whether the therapy led to this endpoint or the therapy did not know. And these are Bayesian decision rules to determine whether or when therapy is with low probability or success should be discontinued, and therapy is high success, allow. So okay. So here if you see, they expect that about 85% chance in the next phase 3. So based on this data of what is going to succeed and what may not succeed, you go to next level of trial in phase 3. I come to this classic study done by recovery is a randomized evaluation of COVID COV therapy. Okay, therapy. They have taken here from ERY therapy recovery. This was a study planned by Oxford Group in I think UK, and by now I think all of us know Oxford Group. They have been very active in the field of COVID. I think our Covishield vaccine is I think kind of a gift of Oxford Group. They were very early in disease to decide that. They also did large study of this nature of recovery, and I think it's worth reading this in detail to understand how they did it. It's an adaptive design. So there is a randomization done of first of all usual standard of care. Standard of care actually was I think left to individual hospitals, but by and large it was fixed in terms of no oxygen and no whatever you do no in basic no antibiotics or this was very early in the game 2020. Now so this is SOC plus antiretroviral. I think that initially I think people were focused on this antiretroviral drug no lopinavir ritonavir no these actually work in HIV. Now the first trials I think published in New England in 2020 showed that this is not effective, but I think there was still a lot of focus on this trial. The second group was corticosteroid, third was our HCQ, and fourth was azithromycin. No? And objectives is a very simple endpoint: effect of study treatments on all-cause mortality at 28 days at the first randomization. So they followed each patient for 28 days. Okay. We'll talk more about it later. A basket trial involves multiple diseases or histologic features in cancer. No, participants screen for a presence of a target. Target participating patients are into trial, and trial may involve many different diseases or histologic features. No, so you see, this looks like a basket. Disease one, disease two, or it could be disease three, or it will be different types of histology. Skin presence of target. Now, target is usually a biological target, and then you give single drug to these people, okay, those patients. So they are also called if you look at extension of adaptive trials, there are multi-arm, multiple interventions, and multi-stage. So you can as I showed you, you can do both. You can have multiple therapies, multiple diseases, and even multiple interventions. So just to come back to the platform trial, which I want to talk in more detail because of recovery. A group of patients, this is a population, you have standard of care, intervention 1 and 2. So you have this, each arm there are patients going on. Okay, interim analysis based on the outcome. So if this arm does not work, so you stop the further enrollment of trial here. If this has worked, you continue further on, and you can always introduce new arm here once you feel there is something new has come. So intervention 3 comes up here. Okay, you can even introduce new arm here once you have some data, because you can start adding more interventions. So the trial is perpetual; it continues. No, so ineffective arms are dropped, effective arms are continued. For example, here this intervention probably is not working, so they've stopped it here. So recovery, this what I told you about the Oxford trial was done in this way. They took 18 years and above adult patients admitted to hospital, proven or suspected SARS-CoV-2. They were randomized to, this is a standard of care, no additional treatment. The lopinavir group, dexamethasone group, hydroxychloroquine, and then all of this were outcome. Okay. All cause death and secondary duration of hospitalization and need for ventilation. And see how fast it moved. No, this is 100 days of trial. In fact, I believe they started the studies in March 2020, and the first data of this dexamethasone came very early in June, July. See this. So 31st January 2020, first UK case confirmed cases. They decided to start the studies see in March 2020. I remember 20th March we had this Janta curfew in India. But before that, they started this study in UK. 5th March, first death occurred. 13th March, they submitted for ethics committee approval. So within, if you see, they were ready with the draft of protocol. And then they got approval within 3 days. This is very important. I mean, no, in India, I think we say we have taken speed in clinical trials, but I think you see here, within 6 days of giving the approval submission, they got the approval. And obviously, this was reviewed, and not just approved on paper, Then they got ethics committee approval for all the studies. First patient entered on 19 March, okay, and 23rd March became a national lockdown here. You see, by April 3rd, there are 1,000 patients enrolled. By 14th May, 10,000 enrolled. And see, 5th June, first results announced. no clinical benefit from HCQ treatment. So they first said this, so they dropped this HCQ arm, no, and then 16 June they announced that dexamethasone was announced, and that this became a standard therapy in I think UK. So it became no part of the therapy, in June itself, no, and the publications came that time. Okay, and they also have other arms in this. So azithromycin arm was dropped. They also had I think the second trial on plasma. Along with that, they also trial on tocilizumab. All these trials were running parallelly, and they have been publishing results for last 2 years. This is just an example of what they found in dexamethasone data in recovery trial. This is the control group, usual care. You see in this group, 13% did not require oxygen, while here it's 17%. So relative risk of not requiring oxygen was better in the dexamethasone group. No, oxygen only here more required every 1 in 4. Here it was 4% difference. When ventilation again, if you see 40% required ventilation here, no, while only 30%, no. And overall, you look at all participants, and overall the beta were better in this, no. Okay. But you see how fast they got this 1,000 patients data of COVID. That is very interesting, very important, no. And the control arm had also a large number of patients, no. Even today, I'm yet to see a trial of 500 or more patients in India on COVID therapies. We don't have any trials, about 100, 150, that's all, no. And most are done with uncontrolled, bias, not randomized properly, not blinded properly. Okay. And again, coming back to the challenges, it's not that everybody can carry out master protocol. For example, I mean, the other day I was thinking while talking to Dr. Kindu that theoretically, I'm saying theoretically, Ayush could have done a master protocol, no, of all Ayurvedic therapies which are potentially effective for a condition like COVID. So instead of doing somebody doing 100 on Chyawanprash and 100 on Ashwagandha, maybe they could have done 10,000 patient study, with one control group, and the rest would be, comparing different therapies, we would have had adequate numbers to judge what happens. Today, what has happened, with 100, 200, most trials don't appear positive, and then we all wonder that why did we waste so much money and time? Well, this is also true of allopathy. So it's not that the herbal therapies are not effective. I mean, it was a challenge of the disease which is so difficult to treat. Right? But to do this, a master protocol requires a lot of discipline. Trial network with infrastructure. See, in UK, all these centers, we know about 200 centers, they are already trained in doing such studies, to streamline trial logistics, because you have to do everything in the same way. It's like a army unit working all over India and having the same discipline. Improve data quality, facilitate data collection and sharing. Common protocol that incorporates innovative statistical approaches to study design and analysis, enables broader set of objectives to be met more effectively than be possible in independent trials. So this is a kind of discipline of planning a master protocol. If unless you do this, you cannot get the results. So how do you organize it? Infrastructure, you can have a common screening platform for biomarker identification for at least cancer trials. Governance, see, there are committees. The steering committee, overall overarching committee, adjudication committee. Whenever you see a result or outcome, the outcome is not based on what the investigator feels or what comes up from the statistician. The committee, a special committee is we look at each and every patient data and accept the outcome or not, because one might be biased even in that kind of assessment. Then the data monitoring committee, which is going to look at interim data throughout the trial and make a judgment about whether the trial should continue or not based on the safety or the efficacy data. And there'll be one IRB or ethics committee for the whole trial. We require trial networks, clinical centers with experience, processes, randomization, data and safety capture and management. So here I think this will be a common web-based clinical trial data entry, which will be done by all the sites at one point, at a time. Now, let me tell you that we tried from the society where I was working, ISCR, a group of people who are doing data management and statistics. We tried to help the academic centers in such a thing. So we tried to help them design a common protocol and a common platform to enter data. And we thought that we would have got some data. And after 3 months, when we followed up, we found that we had some data of about some 50, 60 patients. And again, it was not complete. So we couldn't do much of this. So that's the challenge of India. Then in India, these trials are difficult, not because we don't understand or we don't have decisions, It's more of no our standard problem of everybody is a leader and nobody is a follower. No, so with a team approach, somebody taking leadership and organizing, it can happen. No, the quality control oversight. Trial design, you have these features, adaptive randomization, other features, long-term modeling treatment probabilities of success or failure, shared control patients, natural history cohort, and biomarker qualification. Okay. So any questions on master protocol?
Uh, Yes, sir. Sir, you were talking about that such a master protocol could have been done for Ayush interventions, so as we can test multiple interventions, and in interim analysis we could uh, modify or take the successful treatment ahead. Uh, Sir, how this was done in the recovery trial, and how interim analysis was uh, really planned? What were the assumptions and primary outcome measures?
Oxford was the main kind of center. So Oxford, I think, designed the protocol. They were the governing the whole trial. They were the making decision makers for the whole trial. Patients were taken from all over. I mean, virtually every center in British, no, I mean, every hospital in UK could take part in the trial. They had made a very centralized no website. Anybody wants to take part in the trial, you can do it. So obviously, you'll have to first get all the approvals. So every hospital had a choice of no entering this trial, and and the whole process. I think no, if you go to their website on recovery, they know they have multiple no YouTube videos, they have multiple no website, no training. So they trained all the sites in the way they should do a study, and each of them had a choice of getting one arm. So when you can't decide the arm, so depending on the next patient which you have got, you will get a number, and that arm you have to start using it. So what will happen is that okay, but they were very clear that the arm should be available in the hospital. For example, for example, if some hospital doesn't have HCQ, then they cannot take part in that arm. No, they may have to take part in the other arms. So it was like no, whatever was available in the hospital was used. No, not no, they did not try to send samples, supplies no from one center place, because that would be no kind of very that would have spent so much time and effort in just distributing the drugs. So each of them could take part. So their main observers was, they'll have to know enter the patient number, the consent data, and the basic demographic data, and then they'll randomization number they'll get. They'll be told to give the particular therapy in the particular dose. So there are different groups planned. The protocol is quite elaborate on this. No, the standard therapy is left to the hospital, but they'll have to kind of capture what standard therapy is done, and then the data of this 28 days of primary outcome of hospitalization or death, they'll have to enter the data in again the system. But this will be ongoing. So they can say, for example, if I am doing a study and I want to add, let's say, about 50 patients, what is the tendency in India? कल हम लोग data enter कर देंगे. कल नहीं, today when the patient is suffering from outcome, patient is discharged or patient is died, that's the time you enter the data, because they are actually already collecting data and accumulating. As the data accumulates, they go on analyzing. So for every arm, they would go on analyzing the data, not with every patient, but they would have got some kind of plan. Okay, let's say, like for example, in dexamethasone, you saw when there are about 2,000-3,000 patients into two arms, they analyzed the data and found out that the dexamethasone really works, because they don't want to wait till then. So this trial is still ongoing. No, I mean, after they finished with dexamethasone positivity, they said HCQ is not positive. Then I think SMS is not positive. Plasma is not working. Finally, I think in that study, remdesivir is also not working. No, in that study, so that the way they've looked at it, it's not working. But the whole study, and then they go on publishing this data as and when it happened, no, very fast. So that's the way the whole thing. But the control was with Oxford, no, in terms of designing, planning, analyzing, deciding, taking decisions. Yeah, there are committees outside to give them advice, but the whole thing was done in one coordinated fashion. But every hospital has the only option of entering the trial, and they would get only kind of no credits for that. I mean, some of them, I mean, you will not get your name on the final paper because you are only contributing the patients. But that's the way the trial is done. No, so that would require a, for example, ICMR in India can virtually do this, but the problem is no, ICMR has a different kind of problem. I mean, the way now the whole thing functions, no, it takes a lot of time, no, and they have a lot of biases about how to do things, no, and then no, lot of preferences. So frankly, they are not able to do it. No, that's a problem. No, otherwise I think we could do it in India. But unless, and that would require no ready centers all over India. I mean, even today, I don't think, I mean, you and I are in clinical research and I have been part of this industry. We talk about no, India has done so many trials. And if you look at CTRI, we have about no 2000 or 3000 or 5000 investigators. But I'm not sure all those 5000 people have the same training and infrastructure for doing good study. That's a problem. No, I can tell you that no, that we know the differences between centers and centers, and it's not that a Mumbai center is better than Delhi center. No, it sometimes happens that a center no in Belgaum is better than no a center in no Mumbai. It's possible. No, because our problem is, I think, as a country we don't do research and we are not disciplined to do research. That's where the challenge is.
Thank you, sir. I think there are many other questions or comments that we can take up after the.
Should I continue or?
Yes, sir. Please continue.
Okay. So the last part is actually very easy because last part is not a new design of clinical trial, but new practical way of managing a trial. And that's called digital trial. You might have heard of this, and this is all now part of this. I mean, this is a self-help hospital. You diagnose yourself and treat yourself. I mean, so this cartoon indicates in a way that in digital trial, patient is the center. So what has happened, if you remember in clinical trials, those who are working in COVID, I mean, at least I am part of this industry, so I know that patients were not willing to visit the hospitals. So how do you manage? This is actually talking about all trials, not just COVID trials. Any trial you are doing. So at that time also India, we are doing about 100 to 200 trials in different indications. And the question was, if patients don't go to hospitals, what do you do? One, of course, is observing, okay, the trial stops, the patient stops therapy, and you forget the trial. So that's one easier option, but that's not happen. At the site, investment site, personal staff safety, screening, enrollment, all this was, I think, suffering because people don't come to hospital. How do you do screening? How do you enrollment? How do you do consent? Ethics committee had similar problems. They were meeting virtually, but they could not ensure all this. And sponsors had worry about continuation monitoring. I mean, even how does a monitor go to a trial to do monitoring of a trial? No, how do you supply initial product? No, so this is what was the challenge. So this came up around that time. Although I think this is again in literature for quite some time. I mean, there are multiple definitions we will hear. It's called virtual or digital trials. Now, virtual is not the data or the patient. No, the patient is still there. No, the data are still there. Site free or remote trials activities performed without visits to hospitals. So this is the basic theme is that patient will not go to hospital for the clinical trial. The hospital will try to handle everything at patient's home. That's a main kind of big change here. Home based convenience of performing some trial functions at home. Some functions. See, if you can't do anything at home, okay. Dare to participant. Central unit communicates with and sends treatments adapted to the participant. So here I think, this is one thing which has to be done, that if you are going to do a trial, a home-based trial, the therapy for the clinical trial, which is all blinded, randomized, all that has to come to patients at home. Okay. Virtual communications and assessments are performed using digital technology. So obviously it's going to be a use of telemetry, mobiles, kind of websites, portals, all that. Okay. Decentralized, hybrid, and decentralized. Use remote data collection because you are not, the patient is at home and you are collecting data. Virtual communications, emails. You can even have WhatsApp. You can have social media. Okay. Home-based services to replace some in-person visits. So this is actually a new thing which happened. Obviously, this was more a kind of a need to do this because otherwise the trials would have stopped. So in India also, most companies started doing this, but obviously you can't do the whole trial as it is, virtually. So how is it done? Typically, let's say if KEM is doing this trial, the KEM hospital in Mumbai will be the center, but the people are not going to KEM. KEM will send staff to their home to collect the data on everything, right from selection, consents, giving the therapy, collecting blood samples, all that is done at home, and the data will come to KEM. So assessments and treatment performed without a clinical trial site. Investigators and staff located at the coordinating center. Participants may establish relationship with staff by telephone, email, video. So there are different options available. For example, if you want to see a patient, you can see it on a video, like doing the telemedicine approach. You can check uh, their symptoms on telephone, or you can even ask for email for. So the consents could be done in multiple ways. All trial functions are managed by the equivalent of coordinating center or contact resources. You might have a CRO. Okay. So what is the benefit? Now, benefit of the patient is actually patient doesn't have to go to the hospital. Now, well, you might find that in India people want to go to hospital because they want to meet. But some situations, for example, elderly, with advanced disease, it's so tough to take them to hospital. So there are smaller, clinic subunits of clinical trials, where I think this is really a boon for the patients. And since you don't have to go to hospital, there is a better chance of more people getting part in. Because today, if you tell a patient who is let's say working person, who is employed, that you have to come to hospital every week, they'll say, oh, I can't take part in a trial. But if you say no, we are not going to, you don't have to come to hospital. Maybe every Saturday, we will call you up, through, let's say, a video call or telemetry, and we'll take your data. He said, okay, I don't mind taking part in a trial if it's no good trial. So patients have better recruitment, retention, access to patients beyond physical proximity. Now today what happens, for example, I told you about KEM. If KEM wants to do a trial in Mumbai, it will be only maybe in the Parel area, where they are, maybe let's say up to all of Mumbai. But theoretically, a KEM hospital can handle a trial of patients in Pune also, because you don't have to be physically there. Reduce cost by simplifying the protocol and substituting less expensive assessment. So this, of course, is a kind of a dream for everybody, but this may not happen because most trials are complex. Recruitment, as I told you, not limited to one site. You can recruit from any source: social media, online patient communities, from electronic medical records, clinical encounters, camps. People interested in study refer to a study website or central phone number. Now, for example, I remember that during this COVID time, the vaccine trials are going at many centers, and the problem was people are not getting enough people to take part in vaccine trials. Now I don't know. That time if the company and the sites had thought of that, okay, we will arrange to give them vaccine at home and also follow up at home, maybe more would have taken part. At least you could have done a lot of studies, even in people who are elderly or immunocompromised. Data entered directly by participants online using secure systems. So patients will have to be trained to enter data. That's a big challenge for country like India, that we don't have patients who are literate enough to even use smartphone. Data collected from mobile services and wearable sensors. Today, for example, all of you know that you can measure pulse rates, blood pressure, all that through devices. If the patient can take part of it, okay. No source documents at sites to review. So all the data will be at the patients only. Okay. Informed consent. This is going to be a challenge because it's not going to be a standard paper-based taken consent in presence of the investigator. So you'll have to have a detailed SOP of how do you take consent. And I think those who are very keen, you can check the FDA's guidance on how to take consent in this situation. So it's online e-consent. You can ask questions by telephone, text, or email. And I remember that some effort was done by some people in I think Ayush studies. I think I remember one prophylactic study where I think they made a mobile based consent, and also some small form to check the symptoms. Well, I don't know whether they were done in the right quality, right ethical way, but there was an attempt done by some sites to do this, which was a good progress. For treatment carrying more than minimal risk, conversation with health staff is required. So obviously, you cannot take consent for all just like that. You might have to send a doctor to their home to make them understand what are the risks. Electronic signature compliance with regulation. Now, this is one thing which we are not yet doing in India. We are not yet doing routine. There is a way to get electronic signature certified, which is a legal acceptance. And finally, written signature is sent by photograph from a cell phone scan. So there are multiple ways of getting a signature of this patient along with the consent. How do you examine them? Well, some things are done on telemedicine interview, examination. Limited examinations in home by resident nurses. So some things may be at home. For example, if you require actual blood pressure, pulse, and some data, you may have to do it. Portable in-home test. Today I think oximetry we all got used to measuring oxygen at home. But for the spirometry, there are portable machines available for even pulmonary functions now. And obviously, for images, patient will have to go for let's say MRI. But patient doesn't have to come to hospital all the way, for MRI. Patient can go to nearby center, and the kind of the image can be sent to the central body. Again, collection of sample at home. This is, I think, not new. I think all of us got used to it, that you can order a home test. Okay, you can even include some special things which can be done at home, but I think our homes are not big. Samples go to a central lab, and obviously some things can be tested at home. For example, you can do blood glucose testing at home. This is the area which will require some amount of doing, but I think you have to have oversight of adverse events. Participant reports adverse events to coordinate center attended by form. So adverse events are recorded directly 24 by 7. Medical care of these events you require no local source of care. So obviously either the hospital will have to send people, or you might have to tie up with a local general practitioner or a physician who can provide care for these events. Okay, and this will have to be kind of this records medical records have to be reviewed the patient's consent to obtain records so you'll have to do more work on this part of that consent. Again, I think if you are sending the samples, so the treatment meant by central pharmacy. Oral or topical treatments are easy to ship to a patient's home because of it's very easy. But parenteral treatments will require some amount of thinking that whether can you do it at home or not. Because some things are possible, and for this again, I think you might require research nurses, But again, you look at everything that you do. For example, here authorization by the physician, conversion identity, and delivery. So you have to have documentation of the patients getting this treatment. But this is not going to be easy, and I think I personally feel that in country like India, it may be very difficult. So maybe in situations which are after the drug is approved and marketed, maybe you can do some studies which are maybe in the situation of marketing or real world situation you can do it. But otherwise, for example, ethics committee oversight. I mean, how will the ethics committee ensure that the patient's rights are protected? Consent. Do we accept this home-based consent, e-consent? India, we are not yet accepting it. Internationally, it is already accepted. How do you provide medical care at home for a serious adverse event? And can the company access directly patients? Because this is going to happen that the sponsor is involved and it's going to be in the trial. They send drugs to the patient's home. They send laboratory to home. Now, there is a way that patients, the company might come to know who is the patient. That's where the problem is that in clinical trial setting, although there are patients, but the company doesn't know which patient and who is getting what treatment, and there's supposed to be a confidentiality issues of that. Technology, I think there's a lot of mushroom of variety of apps and media in India, but I think I doubt whether they have validity confirmed. We talk about so many apps, but I don't know whether those apps are really valid in terms of what they're supposed to do. And main problem will be that for our patients who are largely illiterate and uneducated and maybe poor, they can't afford a smartphone, and you have to train them in the smartphone. I mean, it's possible. I'm not saying not possible, but today I think it's struggling. So even that's why even today, I mean, I don't know how many of us are using digital payment modes, in spite of no possibility of doing this transfer in COVID. Practical, I feel that patients are not happy not meeting their doctor. So I think in Indian setting, even if you want such a trial, I think the patients will be very happy to go and meet the doctor. They don't trust the technology so much. Okay. Plus quality of cost of hospital versus home visit now. I mean, see, this is the what we don't know. I mean, when patient goes to hospital, there's no additional cost for the hospital. They anyway are looking at so many patients, but now we are sending people at home. The doctor, the nurse, the pharmacist. There are complex endpoints and assessments. You do require people to visit the hospital. IV administration. Then how do you store things at home? I mean, it's assumed that you do only standard estimations, lab estimations, but something special. How do you do that? For example, you have to store the drug at 2 to 8 degrees. Many of the drugs are like that. So how do you do that? How do you ensure compliance to treatment? And how do you ensure remote monitoring? The monitoring which is done by the company, do you have to do this monitoring in this setting? How do you do that? So finally, to conclude, post-pandemic paradigm, there are opportunities for doing clinical trials in a different way. Real-world data to develop hypothesis and supportive evidence to do ultimately clinical trials. Large-scale national and international clinical trial master protocols essential to generate high quality evidence rapidly, and patient-centric trials balancing ethical, technical, and practical challenges. So that's the last slide, and we can have discussions, right?
Sir, thank you very much for a very rapid overview of newer designs such as master protocol platform approach, and nicely elaborating the example of recovery trial. Also, you also discussed many interesting aspects of utilizing several digital platforms and dig- digital trials. Sir, do you think that newer methods such as the platform trials, the flexibility of protocol or master protocol, and involvement of several sites, large numbers, multiple interventions can lead to diversity or variations in data, and that may also lead to some of the challenges.
I mean, as I answered that, see, the whole trial is designed in a way that you reduce the diversity in data and the endpoints. That's the purpose. That's why you require discipline of a common protocol, discipline of a team, and discipline of statistical work done by a common. So see, when you say it's not like 100 sites will do 100 trials. Only one trial done by 100 sites, and those 100 sites give everything to the one site which is doing the trial. So unless we accept that kind of kind of pyramid approach and discipline, we cannot do a trial. See, that's why in India we are not, we can't do a 2 or 3 or 5 center trial, but we don't have a teamwork approach. No? Who's going to lead the trial? No, who is going to manage it? No, so that's a problem. So talking about platform trials, no, in India without a background of large number of equal standards centers. I'll give some experience of how things happen. I was on BIREX committee to BIREC wanted to develop new sites, but they feel that as we go forward, this is a depend on biotechnology. We go forward, will require let's see. Let me just try to. Is my screen now stop? No, I've stopped. No.
Yes, yes, screen is now not stop.
Okay. So
Okay.
coming back to this issue of BIREC, so they asked to large number of hospitals from all over India to put up a proposal of how they will develop a clinical trial center, and then it will be mainly for doing drug development and supporting India's support for developing new drugs, maybe generics. Now we heard from almost about 10 or 15 leading hospitals from all over India. This was actually screened, they were screened from different proposals. We found that except for one, which was obvious, Tata Hospital, we said we have done this and we can do this. Everybody else was asking, I want space, I want laboratory. Now, I think idea was not to give space or laboratory. Idea was that if you are already doing something, can we upgrade you to do a proper trial? Now, I think I found even that in discussions, when we asked for CV of the investigators who have done a clinical trial or something, like for example, they wanted to go diabetes trial, but the CV was of orthopedic surgeon now. So the sites did not even know that what is a good clinical trial site. So without that background of quality, see our problem, in Mumbai, Pune, we have seen KM Mumbai, we have seen KM Pune, we have seen Sahyadri, we have seen JCDC, we have seen Tata. So we assume everybody is at that level. It's not. I mean, average clinical trial site is still struggling with no basic issues. So put a firm foundation of training, quality, and all that, we cannot take off and do a master trial. So I mean, the issue of what you mentioned about variation comes because we are still thinking that I will do what I want to do. No, we have a uniform approach of selecting patients, and we will not be able to violate those. Then only it can go. No, yeah. I think there are some questions in the chat box. I think
Yes.
I'll take the QA question first. There are two here.
Yes, please. Yes, yes.
There was a question from Garima Gupta. How insurance related to higher rate of real world evidence? This is not insurance. I mean, we are going to use data collected for medical insurance for doing real world research. So, for example, as I told you about this Israeli study, it's a large insurance group. For example, let's say Mediclaim. We all have Mediclaim policy, and some of us are admitted for in hospital. So, Mediclaim has a lot of data on different diseases. Now, theoretically, that data can be used to analyze and look at certain evidences. For example, in heart failure, one of the challenges now, complications of no admission to hospital and death. Now we don't have this data available in the public literature because nobody keeps it. No, so insurance data can help in generating real world data. Now next is from Amit Nakanekar. Can direct contract of farm company with patients influence virtual trials? See, that's what I mentioned. See, that's a question we have. The company doesn't go to patient directly. The trial is still under control of the hospital, but company will be involved because they are happening at patient site. And I think unless the hospital which is willing charge of it again takes charge of it. For example, if I have to send samples to patient, drug samples, it would not be going from 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 my company. It will go from the hospital. KM will send it to them. I will not send. So there are ways of reducing this chance of interface between patient and the company. Okay, now we have come back to the standard. Is it a question? I don't know. For many trials in India, DCGI wants, it's not one money. It's for all trials, DCGI wants every recording of consent process. And the consenting process seems difficult. So that's okay. That's what I'm saying. No, we are very far away from what I'm talking right now. No. Okay. 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 So that's the only question I see. Okay. Any more questions? Because I didn't see any more questions in the chat box or the QA box.
Sir, as we were discussing just before we actually started today, in case of Ayush interventions or traditional medicine, Ayurveda, we have a lot of observational studies. We have to do a lot of observational studies and studies related to case reports, case series. They are also very important for generating evidence. So, what do you think? What Ayush studies should go with the available real life clinical practice experience, or studies related to studies based on reverse pharmacology?
No, reverse pharmacology is different from this. I mean, you are talking about no. I mean, they are talking about no. I think when you say real world data, we are trying to generate evidence comparable to a randomized trial from real world data. So obviously, your data, for example, in clinical trials, no, we really control data quality by doing rigorous monitoring, designing a protocol, case record form. The sites are kind of going to be really doing it uniformly. When it comes to real world data, I mean, both are in Mumbai, and your in Pune, do they capture the data in the same way? I think at least in allopathy, I know that most hospitals in allopathy have a problem. Public hospitals even today don't capture data as we want it. See, I mean, having two notes in the case record form is not data. Supposing you want to, let's say, I'm just giving an example of let's say you want to talk about let's say a leading herbal therapy for liver disease. Do you have available data in clinical records of their demography, they've taken this therapy and the liver function test? Then you can go further and analyze. But if you don't have the data, we cannot do anything. So that's where the thing, the quality of data, the number of available data, and what endpoints are required. I'll give an example of in KEM, Dr. Shirsagar wanted to study how common is hepatitis due to NTTB. Now what they did, they virtually went through case record forms, which were papers case record, and to find out. Now that's not real world research. No, you can't be looking at each and every case paper retrospectively and getting it. So that success is coming again only if you have proper documentation of the data in electronic form, and then you start to generate hypothesis, because without a question in your mind, you cannot just collect data. I mean, it's only saying that okay, I've got 1,000 people who have taken no, let's say no, Bhui Amal ki. Agreed, but what has happened to those people? No, I mean, so that's where no the challenge is in this center. We as a medical community are still not collecting data enough for us to make even a question. I looked at no CTRA to see how much observational work is going in India. I found about 1,000+ studies are going on in India, which are observational in nature. But out of this 1,000, almost 900 were prospective because they don't have good data. They don't have data enough to do anything. So only about 10% were not trying to do something with the data available. No. There's also a very interesting study published by Dr. Handa from NIMS. They looked at 30 publications on rheumatoid from India, 30 Indian studies published now, and they are all observational studies, no, not clinical trials. In 30, in 10, they did not have data on epidemiology. In 10, they did not have data on treatments. In 10, they did not have data on features. So how do you now go to the next step of even making a judgment of what this is going to be? So that's a problem that we as a country, I think, are not yet capturing data systematically in our practice, clinical practice. And this is true across. I mean, it's not only public, but even private as well. The same problem is there. No, there is, I think, one question here. Are there guidelines or checklists available for platform trials? I mean, see this on no, it is not. Equator network will tell you about how when you publish the trial, no, what is required in this. No, so it's not there. It's not at a publication level. The publication will be like a standard a clinical trial publication. So when you say checklist, I don't think it's there on the Equator for a master. But yes, there are sub checklists for I think newer kind of trials. There are checklists for strobe is for observational studies. So there are checklists available, but I don't think there is specific checklist right now for a master trial or something. But I may be wrong. You can check up.
To move forward with the master protocol and platform studies, those could be very time and resource sparing approaches, right? We should
definitely
have these standardization exercise and systems in place, right? So
yes.
in this scenario, when many institutions, Ayush institutions, they started many studies and people are sensitized for these COVID-19 studies. I think this is the situation where we can have such standardization exercise and train people, those are really involved in studies.
No, but I'll tell you, I think it's not enough. See, when you say standardization, I think that is still just understanding how a good protocol is written and how it is conducted. But I think we are still stuck with the basic question of documentation. I mean, I think I don't want to give you something more, but I have looked at I mean, a lot of studies in COVID. I mean, and including herbal and homeopathy. The whole problem was the same thing, that the documentation was so poor that very often you did not have enough data to make a judgment. And ultimately, when the study is done, the researcher wants to somehow publish it. For example, I remember one study came for review, and they had to talk about is the therapy more effective compared to control. Now they did not have a control arm. What they did, they picked up a newspaper report and said, no, in this city so many were hospitalized, so compared to this we are better. Now that's not the way. I remember one large study being done under Ayush Hospital did not have a control arm, and they were making claims on the study with some 10,000 patients, but there was no control arm. So that's a problem, that we have not understood that some things are given now. I mean, I'm not saying this is not modern way of doing it. There's only one way of doing good science in clinical research. Yes, you can have any therapy. Recently, I was involved in kind of a quality of life study for a alternative system. I don't want to tell you which system. Now the same problem. It has taken us 3 months to make a protocol because they did not know how to put this in the setting of it was add-on therapy, chemotherapy plus this therapy to look at how it works on the quality of life. But for me, it took make them understand that what is all this now about 3 months time. No, you're not understood yet. No, so that is where the problem is at, and obviously documentation done by big companies succeed because they put no tremendous resource no in training, documentation, checking, monitoring, all that, and a lot of money there. But average center is not really so unless you do your own research and start doing in kind of systematic way with quality, then you can easily do it. But we cannot create centers only for trials as it goes by. It's not going to work out that way.
Yes, absolutely.
There
Sir,
is something in
there is a
chat. I don't know
question.
what is it. It's only no, it's I think more of I think YouTube will have the presentation. Okay. Okay. There's one I think QA. I think asking for umbrella trial for Arrow. I think the issue is same. No, either umbrella or master or platform. The view is one can do it provided. I mean, I'm joking. We are in Oxford country, but we should become Oxford country. Now Oxford, I think some of you might have seen that there should be a comes down to know how they did it. It's available all over the net. But the challenge is, do we have a central researcher and multiple centers who are willing to support? I would say it takes time and effort.
Thank you. Thank you, sir. Thank you
Yeah.
very much. Any other
Just
observation?
just the thought. I mean, before I close, I think since the question came up, and I thought, in relation to Ayurveda, for example, you can do, let's say a umbrella protocol with multiple different prakriti, patients, in the same trial, you can have multiple different prakritis, and then you can try to compare one drug or multiple drugs. And that's one idea which I'm getting right now, subtypes. But then Girish knows the challenge of getting this Prakriti Nirnaya done in a standard way, entering them in trial and then blinding it or randomizing. So I think it's a task which is yeah. The problem, but these designs are not observational studies have a problem of confounders. But when you come to RCT, these are all RCTs basically. Only thing there are different designs. So when you say confounders, it's more of a bias of knowing something and taking a decision rather than the confounder of the patient variables. I mean, I think okay. Yeah, Krish.
Yes. Thank you. Thank you very much, sir, for this very elaborate session that covered many topics, and your experience is certainly very helpful, and we will continue this interaction further. Thank you very much. Thanks a lot for
Thank
your
you.
time, sir.
Thank you. Thank you very much.
Namaskar.
Yeah, thank you.